DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions for life. Every living organism, from the simplest bacteria to complex plants and animals, carries DNA that governs its development, functioning, and reproduction. It's the very foundation of what makes us who we are. But what exactly is DNA, how does it work, and why is it so important? Today, we'll explore the incredible world of DNA, its discovery, its structure, and its role in the functioning of every living organism on Earth. The journey into the world of DNA begins with a brief history. In the mid-1800s, scientists began to understand that genetic traits were passed from one generation to the next. However, it wasn't until the early 20th century that the molecular basis of heredity was discovered. By the 1950s, two scientists, James Watson and Francis Crick, famously unveiled the double helix structure of DNA, with crucial contributions from Rosalind Franklin's X-ray images. This breakthrough in 1953 completely changed our understanding of genetics and biology. Their work led to the discovery that DNA is not just a genetic material but a code that can be decoded to understand the traits and behaviors of organisms. At its core, DNA is a very long molecule made up of smaller units called nucleotides. There are four types of nucleotides: adenine, A, thymine, T, cytosine, C, and guanine, G. These nucleotides pair up in a very specific way. Adenine always pairs with thymine, and cytosine always pairs with guanine. The sequence of these nucleotide pairs forms the genetic code, and this code holds the information that cells need to produce proteins. These proteins perform a wide variety of essential tasks in the body, from digesting food to carrying oxygen in the blood. DNA's structure is often described as a double helix, which looks like a twisted ladder. The nucleotides form the rungs of the ladder, and the sugar-phosphate backbone forms the sides. The beauty of DNA lies in its ability to replicate itself with near-perfect accuracy. This allows organisms to pass their genetic information from one generation to the next. Before a cell divides, it replicates its DNA so that both daughter cells receive a complete copy of the genetic code. This process is called DNA replication, and it ensures that every cell in an organism has the same instructions for how to function. But DNA doesn't just replicate itself, it also contains the information needed for protein synthesis. This process involves two steps: transcription and translation. In transcription, the DNA sequence of a gene is copied into messenger RNA, mRNA. The mRNA carries the genetic message out of the cell's nucleus and into the cytoplasm, where protein production occurs. In translation, the ribosome reads the mRNA sequence and uses it to build a specific protein by linking amino acids in the correct order. The sequence of amino acids in the protein determines its structure and function in the cell. This is how genetic information stored in DNA leads to the creation of the proteins that make up and control our bodies. One of the most exciting aspects of DNA is how it can be used to trace our ancestry. Humans share about 99.9% of their DNA with each other, but the remaining 0.1% is what makes each individual unique. By studying variations in our DNA, scientists can trace human lineage and evolutionary history. For example, scientists can track how our ancestors migrated across the globe, from the early humans who first left Africa to modern populations. But DNA isn't just about human traits or history. It's also the key to understanding diseases. Genetic disorders, such as cystic fibrosis, sickle cell anemia, and Huntington's disease, are caused by mutations or changes in specific genes. These mutations can alter the function of proteins and lead to disease. Understanding how these mutations occur can help researchers develop new treatments and therapies. For example, scientists are working on gene therapies that could potentially treat genetic diseases by correcting the faulty genes responsible for these disorders. Advances in genetic editing technologies, like CRISPR, have made it possible to cut and edit DNA with unprecedented precision. CRISPR allows scientists to remove, add, or alter DNA sequences at specific locations in the genome. This has the potential to treat genetic disorders, enhance crops, and even create genetically modified organisms, GMOs, with desired traits, such as resistance to diseases or improved nutritional content. Despite the tremendous potential of gene editing, there are important ethical considerations to address. Editing the human genome, particularly in ways that would be passed down to future generations, raises complex questions about the potential consequences. The ethical debate surrounding gene editing is one that continues to evolve, as scientists and society weigh the potential benefits and risks. Another important role that DNA plays in life is in evolution. Over millions of years, the DNA of different species has evolved through processes such as natural selection and genetic drift. Mutations in DNA introduce genetic variations, which are then passed down through generations. Some mutations offer an advantage to the organism, while others may be neutral or harmful. This process of genetic change over time leads to the diversity of life that we see today. By studying the DNA of different organisms, scientists can trace evolutionary relationships and uncover how species are related to one another. A fascinating example of DNA and evolution is the comparison between human DNA and the DNA of other primates. For instance, humans share about 98% of their DNA with chimpanzees, which suggests a common ancestor millions of years ago. However, small differences in DNA are responsible for the traits that make us distinct from other species. These differences, along with environmental factors, have shaped the development of humans as a species. In addition to evolution, DNA is crucial in the development of an organism. During embryonic development, DNA guides the differentiation of cells, which become specialized into different types of cells like muscle cells, nerve cells, or skin cells. This process is driven by specific genes that control the timing and expression of proteins. This is why every cell in your body has the same DNA, but different cells can perform very different functions, all because of the genes they express. Another important aspect of DNA is the process of epigenetics, which studies how external factors can influence gene expression without changing the underlying DNA sequence. Environmental factors like diet, stress, and toxins can impact how genes are turned on or off, and these changes can sometimes be passed down to future generations. Epigenetics adds another layer of complexity to our understanding of genetics, showing that our environment can have a lasting impact on our genetic health. And let's not forget the role of DNA in the growing field of forensics. DNA profiling is now a standard method used in criminal investigations to identify individuals or determine biological relationships. The uniqueness of an individual's DNA, even among close relatives, makes it an incredibly powerful tool for solving crimes and establishing paternity. The study of DNA continues to unfold new discoveries every year. With the advent of next-generation sequencing technologies, scientists are now able to sequence the genomes of entire species in a matter of days, leading to new insights into biology, disease, and even potential extraterrestrial life. As our understanding of DNA deepens, we'll continue to unlock the mysteries of life at the molecular level. The story of DNA is just beginning, and as we move further into the 21st century, who knows what new secrets we'll uncover about the very code that makes up life on Earth.